A very good morning to all and a warm welcome to you all for 5 days AICTE ATAL sponsored online FDP on Nano Furtherance of Pharmaceutical Engineering, an industrial pursuit organized by Nirmala College of Pharmacy, Mangalagiri. Myself, Srikala K, Assistant Professor, Nirmala College of Pharmacy, Atmakur, Mangalagiri, take the privilege to inviting to you all. On this auspicious occasion, let me take an opportunity to introduce our college. Nirmala College of Pharmacy is a Catholic minority institution established and administered by the Society of Catechist Sisters of St. Anne, Hyderabad, under Article 31, according to the Constitution of India. Nirmala College of Pharmacy started in the year 2004 by the Congregation of Catechist Sisters of St. Anne, Hyderabad, and is offering Diploma, B Pharmacy, Pharma D, M Pharmacy, with 3 specializations, that is Pharmaceutics, Pharmaceutical Analysis, Pharmacology, and also PhD. The institution is approved by AICTE New Delhi, accredited by NAC, approved under UGC 2F and 12B, and also approved by PCA and affiliated to Acharya Nagarjuna University, and secured All India NIRF ranking 76 to 100 band in Pharmacy Division from past 4 consecutive years. The institution received grants of 55 lakhs worth from various governing bodies like AICTE, DST, ICMR, etc. With these achievements, our Nirmala College of Pharmacy has been successful in providing and maintaining high quality in teaching and research in pharmaceutical sciences. Now, it's the time to know the importance of FDP. The Faculty Development Program intends to provide financial assistance to facilitate upgradation of knowledge, skill, and intends to provide opportunities for induction training to teachers employed in various disciplines like engineering and technology, as well as pharmacy, etc. The scheme is designed to enhance the teaching and other skills of faculty and to make them aware about modern teaching tools and methodologies. It provides an opportunity to acquire knowledge about current technological developments in relevant fields. It will not only promote the professional practices relevant to technical education, but also motivate the faculty to achieve competitive teaching and learning environment. This FDP provides a premier interdisciplinary platform for researchers, practitioners, and educators to present and discuss the most recent innovations, trends, and concerns, as well as practical challenges and solutions adopted in the fields of pharmaceutical engineering. Now, let us start our today's FDP program by inviting our distinguished guest. First of all, I would like to invite Chief Patron of this FDP, Reverend Sister K. Maria Sundari Garu, Provincial Superior CSE, Guntur Province, to deliver a keynote address. A hearty welcome to you, Sister.
Hello.
Sister?
Hello, sir. Inputunda? Hello.
Yes, sister.
Okay.
Yes, sister.
Okay.
Sister, we are hearing, sir. Yes, sister, you can proceed. Sister.
Yeah. Yeah. Yes. Can you hear me?
Yes, sister.
Am I audible?
Yes, sister, it is audible, sister.
Yeah. Thank you very much for inviting me. And I'm also happy to know about our college and all the facilities that are provided by Nirmala College of Pharmacy. On this bright day, I wish you all a happy morning. I am indeed glad to know that our Nirmala College of Pharmacy, Atmakur Mangalagiri, is arranging online faculty development program on nano furtherance of pharmaceutical engineering, sponsored by ATAL, AICTE Training and Learning Academy. I am deeply impressed and I appreciate for the hard work and open mind of Reverend Sister P. Alfonsa, patron, Dr. S.K. Abdul Rahman, the convener, and organizing secretary, Dr. B. Pamul Reddy, to make this faculty development program a reality. Earlier, many student development programs were organized by the Nirmala College of Pharmacy. But it is a golden time for the faculty to be part of this seminar to develop the knowledge in advanced technologies, to be researchers. The fruit of this seminar depends not on your just registration, but on your active participation in this faculty seminar. I congratulate the resource persons and participants for their availability in promoting healthcare through nano pharmaceutical engineering in this 5 days online seminar. May God bless you all throughout this seminar. I wish you all the best.
Thank you so much, sister, for your valuable message. Now, I would like to invite patron of today's FGP, Reverend Sister P. Alfonsa Garu, Secretary and Correspondent, Nirmala College of Pharmacy, to give a message. Sister, your voice is not audible, sister.
Sister, please.
Thank you. Respected Chief Patron, Sister K. Maria Sundari, a convener, Dr. S.K. Abdul Rahman, organizing secretary, Dr. B. Pamul Reddy, resource persons, chairpersons, and all the faculty delegates. Good morning to you all. It's a great privilege for me to join the AICTE online faculty development program conducting by our Nirmala College of Pharmacy, sponsored by ATAL Academy for 5 days. I congratulate Dr. Pamul Reddy, HOD and Professor of the Department of Pharmaceutics
for grabbing this sponsorship for 5 days. The selected topic for this program, well advanced technology, which deals in the field of nano products. So all the participants or the delegates who are joining this program, I'm sure will be enriched by the knowledge that you will be getting during these 5 days. So I congratulate all the resource persons, eminent personalities who are initiated to elicit their knowledge. I wish you all the best. Thank you.
Thank you so much, sister, for your valuable greetings. Now, it's the time to invite our dynamic principal and convener of today's program, Dr. Sheikh Abdul Rahman sir, to deliver a message.
Thank you, madam. So, respected Chief Patron, Reverend Sister K. Marie Sundari, President and Provincial Superior, CSA, Guntur Province, Patron, Reverend Sister P. Mansa, Secretary and Correspondent, Nirmala College of Pharmacy, Mangalagiri, Andhra Pradesh. My dear ATAL FDP Organizing Committee members and dear participants, from various universities, good morning and hearty welcome to the online AICTE Training and Learning Faculty Development Program. I am delighted to be as AICTE sponsored ATAL FDP program is held from today, that is 21st June to 2020, Monday to Friday. The title of the program is Delegates and supported to leadership skills. Our FDP provide a variety of opportunities for training and exchange of experiences in design and development of nano formulation, characterization, scale process techniques, upgrade the knowledge in nanotechnology and also obtain the life skills. See that you must attend all the scientific sessions and update your professional skills. Simply don't attend for the sake of e-certificates or any other purpose. Anyhow, our organizing secretary, Dr. Pamal Reddy, will explain the instructions. We thank AICTE for releasing the grants to organize a national level FDP to our pharmacy professionals. My best wishes and heartful congratulations to organizing secretary, Dr. Bhavanam Pamal Reddy. My sincere thanks to our management for their constant support in organizing the nation level conferences, seminars, as well this FDP through online. I also thank our Department of Pharmaceutical staff members for their coordination in ATAL FDP. So I think today is International Yoga Day. Right? My best wishes. Namaste to all. Thank you.
Thank you so much, sir, for your greetings. Now, I would like to invite Organizing Secretary and HOD of Department of Pharmaceutics, Dr. B. Pamureddy, sir, to deliver the opening remarks and instructions regarding AICTE sponsored ATAL FDP.
Good morning, one and all. I am very much thankful to the AICTE ATAL Academy and Nirmala College of Pharmacy to host the faculty development program on nano-furtherance of pharmaceutical engineering and industrial pursuit. I'm very much thankful to AICTE ATAL Academy and Nirmala College for making me as host. I would like to tell you that we have received 200 participants throughout India for this FDP. I'm grateful to all the participants, those who are amazing interest for this FDP. I'm very thankful to all the participants for joining with us. We have a bright sessions in this FDP. I'm giving some of the instructions to all the participants to acquire a certificate from the AICTE at the end of the session. The major important points of this FDP are 80% of the attendance is mandatory for all the participants to get the certificate from the AICTE, and 60% of the marks for the online test, which will be conducted at the last day of the session. So we need to secure the 60% of the marks in the online test. Session attendance link will be shared to the email and WhatsApp. You need to fill up the attendance in the Google form. So 80% attendance will be considered for the participants. And also, participants must be shared the feedback in the personal AICTE portal at the end of the FDP. So these instructions are very important to the participants to receive the e-certificate from the AICTE portal. Once again, I thank the AICTE ATAL Academy and management and principal of Nirmala College of Pharmacy for making me the host for this FDP. And I thank all the participants for joining with us. So the session will start immediately after the inaugural. So attend the sessions and update your knowledge and implement for your career growth. Thank you, one and all.
Thank you so much, sir, for your instructions regarding FDP. It has been our tribute to host all the dignitaries and audience of the program. I'm thankful to all the participants for attending this FDP. We have been fortunate to have the renowned identity from academic, industry, and other areas. I'm very thankful to AICTE ATAL Academy for sponsoring today's program, and I am sure that our college will continue to get significant provision from the diverse groups in the future. Thank you so much for all, and now is the time to start the first session. Now, I would like to invite Chairperson of first session, Mrs. Supriya Ma'am, Associate Professor, Nirmala College of Pharmacy, to introduce the resource person.
Thank you, Mrs. Srikala ma'am, for such a generous introduction. Good morning to one and all present here. Myself, Supriya Chatla, Associate Professor, Nirmala College of Pharmacy. Respected faculty and the research scholars, today is the first day of 5 days AICTE ATAL sponsored online FDP on Nano Furtherance of Pharmaceutical Engineering and Industrial Pursuit, organizing by Department of Pharmaceutics, Nirmala College of Pharmacy, Mangalagiri, Andhra Pradesh. It gives me an immense pleasure to introduce about session 1 resource person, Dr. Sailendra Kumar Mahanta. Dr. Sailendra Kumar Mahanta is currently professor in the Department of Pharmacology and the Pharmacy Practice, Nirmala College of Pharmacy, Atmakuru, Mangalagiri. He is the president of Nirmala Innovation and the Incubation Center and Innovation Ambassador of Nirmala College of Pharmacy, approved under MHRD, Government of India. He has completed his master's in pharmaceutical sciences from Mangalagiri Institute of Pharmaceutical Sciences, Rourkela, Orissa, and sir has been awarded PhD from NIT, Rourkela, Odisha. He has received a research grant for the research work entitled Artificial Intelligence-Based Elucidation of Mechanism of Rifampicin Resistance from Tuberculosis Association of India. He has 2 years of experience as a research associate at Biological Research Division, Glenmark Pharmaceuticals, Navi Mumbai, Maharashtra. He has more than 10 publications in international peer-reviewed journals such as Applied Materials and Interfaces, Biointerfaces, PLOS ONE, and RSC Advances, to name a few. And his research interest includes therapeutic approach for the treatment of neuropathic pain and the cancer, artificial intelligence techniques to find the drug resistance, and protein engineering based therapeutics. Sir has also served as a coordinator for various workshops and conferences sponsored by various government bodies like DST, DBT, etc. He is currently the lifetime member of Association of Pharmacy Professionals, India. See, before inviting the resource person, I would like to inform all the delegates to post the queries in the chat box. At the end of the session, all the queries will be clarified by the resource person. Now, I would like to welcome today's resource person, Dr. Sailendra Kumar Mahanta sir, to start the session on protein nanoparticle synthesis and characterization for formulation and the development. Sir, please welcome, sir.
Thank you, madam. Is my voice audible to all of you?
Yes, sir.
Thank you, madam, for your generous introduction of mine. I thank Reverend Sister K. Maria Sundari, President of Nirmala College of Pharmacy, the Secretary and Correspondent, Reverend Sister P. Alfonsa, providing me the opportunity to be a part of this FDP program. I thank the Principal and Convener for today's program, Dr. Sheikh Abdul Rahman, for recommending my name for the FDP program. I would also like to thank the Coordinator and Organizing Secretary for today's event, Dr. B. Pamula Reddy, for proposing my name for the FDP program. I thank All India Council for Technical Education, New Delhi, and the ATAL FDP program for considering me to be a part of this program. Thank you. Now I will be moving ahead with my session. Can I request the host to permit me to share my screen?
Yes, sir. Sailendra sir, we made it, sir.
Yes. Is my screen visible to all of you?
Yes, sir.
Okay.
Yes, sir, it is visible, sir.
Okay, so my topic for today's session is protein nanoparticle synthesis and characterization for formulation development. Now, before going into the session, I'll just like to know how many of you people know about these protein nanoparticles and nanoparticle related products that are under clinical trials. So before I go into the session, I just have a query to know whether the audience is aware that there is a protein nanoparticle formulation available in the market and that is approved by US FDA. So I will request all the participants to mention the name of the formulation in the chat box so that we can move ahead with the session. I'll request all the participants, if you know any of the protein nanoparticle formulation used for drug delivery and it is approved by US FDA. So without any further delay, I'll move to the answer, and that is Abraxane. Now, this particular formulation comprises of human serum albumin nanoparticle, and it loads paclitaxel for target delivery at the cancer site. Now, this particular formulation is used for metastatic breast cancer, and it is approved by US FDA in the year 2005. And you'll be surprised to know, after the advent of this formulation in the year 2005, approved by US FDA, there has been a series of clinical trials with respect to nano formulations. At present, there are close to 15 to 20 formulations which are in various stages of clinical trial, starting from phase 1 to phase 3, and their results are really promising. Most of the protein nanoparticles are also making way into the clinical trials, and depending on the type of the drug molecule we want at the target site, we can customize our protein nanoparticle. So with this background, that protein nanoparticles are really entering into the market, and it is not the first time we are seeing it. From the year 2005 onwards, there has been a constant effort to bring protein-based nanoparticle formulations into the market. Now, why nanoparticles? Before going into discussing about why nanoparticles, we should know what is a definition of nano. Nano in the sense nanometer. So the International System of Unit represents 1 nanometer as 10 to the power minus 9 meter in length. Practically speaking, anything that can be assigned a unit as nanometer can be considered as a nanoparticle. For example, we can still consider a particle having a dimension in between 1 to 1000 nanometer as a nanoparticle. But there are various institutions and various governing agencies. They have strictly defined the diameter of a nanoparticle in the range of 1 to 100 nanometer. The US FDA refers nanomaterials as materials that have at least one dimension in the range of approximately 1 to 100 nanometer and exhibit dimension dependent phenomena. Now the link for that particular document can be accessed. The link has been provided here, and you can go through that document where they have clearly defined that at least one dimension of that particle should have a dimension in the range of 1 to 100 nanometer. Now, if we closely look at this picture, you will get an idea of what size we are discussing about. So what are the natural things in and around us? We see the ants. The size of the ant is somewhere close to 5 millimeter. We see the dust mite. The size of the dust mite is around 200 micrometer. Every day we comb our hair and we lose so many hair during the process. And the diameter of human hair is 60 to 120 micrometer. So this comparison is being provided to you to have a perspective of what small we are talking about. We are talking about a particle which is 10 to the power minus 9 part of a meter. The RBC size is around 7 to 8 micrometer. We have the proteins, which have a size of around 10 nanometers. We have DNA, which is having a size around 2.5 nanometers. We have silicon atoms, where the spacing is around 0.078 nanometer. That means it is even small than 1 nanometer. So if you consider all these things around us, you will have a fair bit of idea how small we are thinking about and what are the natural things and what are the size of these natural things in and around us. Now, if you say what are the things that are man-made and close to these nano systems, starting from the very simple thing that we find in everyone's table, that is a pin. The diameter of the head of the pin is around 1 to 2 millimeter. The microelectrical mechanical devices or the MEMS devices, you'll be having the dimension around 10 to 100 micrometer. The self-assembled nature-inspired structures, most of them around 10 nanometers. We have got nano electrodes for various purposes, and those dimensions of nano electrodes are around 1 micrometer. We have carbon nanotubes, which is having a diameter of 1.3 nanometer. So if we clearly see, it is not that nanoparticles or the nano dimensions are new to us. We are very close to it, and we have been manufacturing certain things in and around us in that range. And naturally also, we have so many things in and around us which are in the nanometer scales. Now, if you see the scale in between, that will give you a very fair bit of idea of where we are going to work on. If you see from the topmost part up to 10 to the power minus 6 meter, this is the micro world. Micro world in the sense, all the particle dimensions will be in micrometers. You can assign a particular dimension in micrometer. That range where we work on, that is known as the micro world. We also have between 10 to the power minus 7 to 10 to the power minus 9. This is the range we are talking about, nano world, or the world in which we will find the particles or materials in the nano size range. Now, why we require this nano size range? Can't we work on some other size range? Why this nanoparticles is that important? If we say why nanoparticles, all of you must agree that as we reduce the size of a particle, the surface area of the particle will increase. Moreover, with increase in surface area, we have a lot of surface area where we can modulate, where we can functionalize, where we can attach, where we can utilize a lot of functional particles for active targeting or passive targeting. So if you see in the image, there are a lot of functionalization that we can do. If we see what kind of ligands we can functionalize on these nanoparticles, because the particles are very small, and if we attach any of these particles or any of these ligands, we can have a targeted approach at our target site. So what are those molecules? We can attach polymers, we can attach small molecules, we can attach nucleic acids, we can attach proteins, we can attach peptides, we can attach antibodies. Now this antibody functionalization is going on very rapidly. When we want a particular drug at a particular binding site, just functionalize it with a type of antibody, and that antibody should be responsive to the type of antigen at the target tissue. It is just like fire and forget principle. Take a nanoparticle, load it with a drug, functionalize it with the antibody, and just release it through the intravenous system in our blood. Now these antibodies will be able to recognize the binding sites at various target tissues. For example, cancer. You have a specific protein that is highly expressed in cancer cells. Now those things will be functioning as the antigens for our nanoparticles. So just simply prepare a functional antibody that is going to bind with that antigen, and just you have to release the nanoparticle into the bloodstream, and it will automatically go through the bloodstream and go to the target tissue and bind at the target site. So nanoparticle approach is solving a lot of our problems with respect to targeted therapy. Now nanoparticles find applications in a lot of cancer treatment also. Reason being very simple. Previously, we used to use so many chemotherapeutic agents, and these chemotherapeutic agents didn't have a specificity for a cancer cell. Now, you can consider as if you're driving a car being blinded. You can hit anyone on the road. Same thing used to happen with the chemotherapeutic agents when we were using it to treat cancer patients. So it didn't differentiate between the normal cell and the cancer cell. So the drug molecule used to accumulate in the normal cell, drug molecule used to accumulate in the cancer cells. So it used to cause toxicity in the cancer cells. Very good, no problem. But my problem was this nanoparticle was considering problem in the normal cells as well. So how to avoid that? So you are treating the cancer cell, that's fine, but you are also killing the normal cell. So at the end, in the way of eliminating cancer cells or treating cancer cells within the body, we are also targeting normal cells and we are also killing normal cells within the body. So that led to different approaches, how we can target this cancer therapy. And this is the area where these nanoparticles are finding a lot of applications. You know, you want to get the drug molecule inside the cell, but the physiological design of the cell is in such a way, the cell is not accepting the drug molecule and it is pushing it out of the cell, thereby causing drug resistance. Find a way, find a receptor through which you can get this drug molecule into the cell. Best way is figure out what are the receptors on the cell surface, design a respective proteins or the target agonists for that particular receptor, functionalize it on the nanoparticle coated with a drug molecule, then release this nanoparticle into that cell. Nanoparticle will go into the cell carrying the drug molecule through the receptor. It is going to release the drug molecule in the receptor. Nanoparticle will find its way through the lysosomal pathway and it will go out of the cell. I'll come to it. Why protein nanoparticles? Because there are a lot of nanoparticles, nano devices available in the market. And now the biggest problem of all these nanoparticles is accumulation within the body. Now, why I'm talking about these protein nanoparticles, we'll come to know about it in the later slides. But at this point of time, you understand, if you're using a metal nanoparticle or you are using any inorganic particle, you're using silica nanoparticle, you're using any other type of nanoparticles, there is always a chance of accumulation within the body. For example, a very simple example I'll give, ferromagnetic nanoparticles. Accumulation of these particles in the body can result in hemosiderosis, that your tissues will become fibrous in nature. Flexibility in the tissues will be lost. On the longer run, you cannot move your tissues frequently. So there are a lot of problems associated with other nanoparticles, metallic as well as non-metallic nanoparticles, which have a problem of accumulation within the body. Next is surface charge. Due to the charge present on the nanoparticle, it can remain in suspension or it can aggregate in suspension. If we know the zeta chemistry of a particle, if there is too much of negative charge on the particle and two such particles are close to each other, there will always be the repulsion among them. But if one of the particle is having a positive charge, another particle is having a negative charge, there will be attraction between the particles and that may result in aggregation. But if you see the positive side, we can utilize this function for surface functionality. You can attach different functional groups depending on the charge, adsorption on the surface of the nanoparticle, that too based on charge. You can modify the surface chemistry. You can change the hydrophobicity. You can increase the hydrophilicity. So by just modulating the surface chemistry with the help of various chemical substances, we can attach a lot of drug molecules. We can specifically attach different chemical substances that will increase its T-half in the bloodstream, and we can formulate these kind of nanoparticles. Apart from that, as I told you, we come to know about various types of nanoparticles. We have got quantum dots, we have got iron oxide nanoparticles, we have got silica nanoparticles, we have got nanoshell nanoparticles, liposomes, micelles, virus-based target system, dendrimers, metallic nanoparticles, all these things are there. But being protein is very, very special. Now, I don't say these nanoparticles are going to cause you more problem. There are certain conditions where being a protein nanoparticle won't help you, but being an iron oxide nanoparticle is definitely going to help you. There are certain conditions, particularly laser guided therapy, particularly magnetic energy guided therapy. In those cases, iron nanoparticles are very, very important. But again, as I said, when you go for metal, non-metal nanoparticles, ceramic nanoparticles, the chances of accumulation in the body becomes very, very huge. Now, when accumulation occurs in the body, definitely toxicity will be there in the body, and that is one of the downside of other category of nanoparticles. But definitely the positive aspect of these nanoparticles are more compared to the negative aspects. Now, whenever we say about a nanoparticle, we have already defined the size at around 1 to 100 nanometer. Now, within this 1 to 100 nanometer, we can have a different shape and sizes. It may be a spherical nanoparticle, it may be a cubical nanoparticle, it may be a rod-shaped nanoparticle, it may be a triangle nanoparticle. Not only that, there may be a surface rigidity. That means your particle is very rigid in structure, not easy to degrade. There may be roughness on the surface of the nanoparticle. Again, that may lead to toxicity. Porosity of the nanoparticle is also very, very important, because these are the areas where you can capture the drug molecules. Once it is reaching the target site, from these porous molecules, the drug can leach out and the nanoparticles can go outside the cell. So all these properties of these nanoparticles are going to be very, very important for us. The second most important part is site-specific targeting that can be achieved by attaching targeting ligands to surface of the particles. As I always told you, you have a nanoparticle, you want to get it inside the cell. Simply attach the necessary ligands on the surface of this nanoparticle. As I explained you about antibody, there will be antigens present at the target site. Let it be any protein, design an antibody for it, functionalize onto a nanoparticle, just release it into the body. The nanoparticle will go into normal cells also, but it is not going to attach to that target site, just because it is specifically designed to a particular antigen that is available in the cancer cell. So even though the nanoparticle goes through the normal cell also, it is not going to create any problem in the normal cell, and it will simply go out of the normal cell. It is going to make its activity present only in cancer cells. We can functionalize the surface with the help of peptides. We can functionalize the surface with the help of aptamers. We can functionalize the surface with the help of small molecules or drug molecules. We can release them, and the drug molecule will be released exactly at the site of their action. We can make polyethylene glycol functionalization on the surface of this nanoparticle in order to increase the half-life in the bloodstream. That also we can do. So with this functionalization on the surface of the nanoparticle, we can achieve a lot of formulation benefits. And because of these formulation benefits, nanoparticles are really pursued nowadays. And most of these nanoparticles have already entered the clinical trials, and around 15 to 20 nanoparticle-based therapies are going on in clinical trials at this point of time. The system can be used for various routes of administration. Now, as I told you about an example of just an intravenous system. You can use it through oral route, you can use it through intramuscular route, you can use it through nasal route, whatever route you want, you can give it. Depending on the target site, you can modify the route of administration. Now, these nanoparticles can be customized depending on the patient also you are treating. You want to treat a patient who is having irritable bowel syndrome that is mostly localized in the colonic region. So for that, you need not be giving all the drugs through the oral route, and the drug has to pass through the stomach, through the small intestine, and finally reach the large intestine or the colonic region where you have the target site. Simply design a nanoparticles that have high functionalized to the lumen of the colonic region and give it through the rectal route. That's simple. So depending on the type of route where you want to target the target tissue, you can prepare or customize your nanoparticle. You can deliver small molecules, you can deliver nucleic acids, you can deliver proteins, you can deliver reporting agents. Reporting agents, for example, you can get a fluorescence reading out of it, you can have a radiation reading out of it. Those kind of things also can be done. You can have intelligent release systems. Now, what are these intelligent release systems? For example, you have designed a nanoparticle. I'll just give a simple example for a cancer tissue environment and a normal tissue environment. In a normal tissue environment, you will find the pH level as close to 7 as possible, and close to 7.4, 7.3, 7.2, in that range you will be having. But in case of cancer tissue, you will be having the pH level somewhat low. You can find it in the range of 6 to 7. So anywhere, depending on the condition of the tissue, you may have a variable pH range. Now, you can functionalize these nanoparticles with drug molecules in such a way, these cross-linkers will break at a pH between 6 to 7. That's it. Functionalize it with a drug molecule, release it into the bloodstream. Nanoparticles will move throughout your bloodstream. They'll find a place where the pH is low, that is in the range of 6 to 7, that you'll find in the cancer tissue, and the nanoparticle is going to release the drug molecule then and there. The nanoparticle is not going to release the particle in places where the pH is alkaline or it is neutral, close to 7 or above 7. It is not going to release it. So you can choose the type of cross-linkers that break at a certain pH, and you can target the cancer tissue and release the drug molecule at the target site. It may be pH dependent, it may be charge dependent, it may be dependent on certain concentration of certain physiological molecules present within the cell. And based on that, we can make an intelligent trigger mechanisms of release of drug molecules within the body. Now, why nanoparticles? Again, they control, sustain, release the drug during the transportation at the site of localization, organ distribution, drug and subsequent clearance of the drug. And they can travel very fast, travel through membranes, transport the drug across membranes with less amount of side effects. They can get controlled release. Previously, we used to dose whenever chemotherapy used to be given to cancer patients, whatever concentration of the drug required at the cancer cell is also equal to concentration of the drug present in normal cells. So what it did, it treated cancer cell, that's fine, but it also killed normal cells. So that was not our purpose. Our purpose is to get targeted release, and targeted release along with controlled release. When you release exact amount of the drug that is required at that particular cell, that amount of drug should reach the cancer cell. So unnecessarily, you are avoiding the drug concentration in the total body. Just load the nanoparticle with the desired amount of drug you want at the target site, release it with functionalization so that it is going to target the cancer cell. The nanoparticle along with the drug molecule is going into the cancer cell, releasing it, and then coming out through the lysosomal pathway. Drug loading capacity is also relatively high because of the increased surface area due to the reduced amount of particle size. You will get more amount of surface area, more amount of loading capacity of the drug molecules. But at this point of time, targeted release of drug molecules. Now, coming to the main question, why protein nanoparticles? We have discussed how nanoparticles can be beneficial to us. Now I'll discuss why protein nanoparticles are going to be the best option for us. So, protein nanoparticles are biodegradable in nature. You figure out any nanoparticle, take out silicon nanoparticle, take out iron oxide nanoparticle, take out titanium dioxide nanoparticle, take out zinc oxide nanoparticles. Are they biodegradable? You can say we can make an arrangement for their biodegradability, but definitely that is going to take some time. But protein nanoparticles core structure is made of protein. Protein will break down into amino acids. Amino acids will be again taken up by the body. So it is a win-win situation when you are using protein nanoparticles. You use it for drug delivery. Drug delivery is done. What happens to that protein nanoparticle? It will biodegrade. It will biodegrade into what? Amino acids. Amino acids are required by your body. Amino acids will again be taken up into the system and it will be utilized for preparation of new proteins. So it is a win-win situation when you are using protein nanoparticles. They can be easily metabolizable. You can metabolize them very easily. You can degrade them very easily. You have the enzyme systems within the body that can degrade these protein nanoparticles. You have the proteasome degradation pathway present within the cells, which can degrade these protein nanoparticles and they can eliminate all the amino acids for further usage within the body. Moreover, they can be easily manipulated. What type of manipulation we are talking about? Surface hydrophobicity. You can manipulate the protein nanoparticle surface hydrophobicity. You have a drug of interest which is hydrophobic in nature. You want the protein nanoparticle to be hydrophobic, having a hydrophobic surface. You can do it. There are different experimental techniques are there where you can address the hydrophobicity issues of the nanoparticles, increase the surface hydrophobicity, allow both the drug and the nanoparticle to come to each other. Proper adsorption will be there, and you can target it for the drug release at the targeted site with a suitable functionalization. Now, all this, apart from this, you can make a covalent drug attachment also. Now, all of you know, covalent drug attachment is one of the strongest form of drug attachment you can do with any other target site. Let it be receptor, let it be protein, let it be whatever functionation you are doing. If you do a covalent drug attachment to a protein nanoparticle, no matter what you do, unless and until the required conditions are present for the release of that drug won't be released. Now, that is the beauty of this protein nanoparticle with covalent drug attachment. Where you want, when you want, what amount you want, you can release the drug with these protein nanoparticles. Now, protein nanoparticles come from various sources. We get it from animal source, we get it from human source, and we can get it from plant source. Can we use all these protein nanoparticles physiologically? My answer is no. you have to tweak with the protein structure, you have to tweak with the protein's immunogenic problem, and you can use it for your physiological benefits. Like animal proteins, you cannot use it directly. You have to reduce the immunogenicity of the proteins. You have to improve or make it look as similar as to human proteins so as to bypass the immunogenic recognition by the body. Same thing with plant proteins also. Now, the best solution to all this problem is you should always use a human proteins. What kind of human proteins? You've got human serum albumin, you've got lysozyme, and there are a lot of proteins present in human body that can be utilized for preparing these protein nanoparticles. Now is the time for recombinant DNA technology. You wanted insulin within our body, you got it from E. coli. If you want human serum albumin, that also you can go from get it from various organisms. And right now, whatever human serum albumin nanoparticles are coming into the market, it is through recombinant DNA technology. So my tip for this choice of a nanoparticle is always try to prefer a human protein for preparing a nanoparticle. Get it prepared through recombinant DNA technology. Prepare the nanoparticle. Since it is going to be very close resemblance of a human protein, chances of rejection by the body or immunogenic reactions within the body will be very, very less. But all the time, this particular option is not available. So we have different sources of proteins coming from animal sources as well as plant sources. And the very common sources of animal proteins are bovine serum albumin. You've got bovine alpha lactalbumin. You've got lysozyme. You've got zein. Zein is coming from plant source. Lysozyme, bovine alpha lactalbumin is coming from animal source. Now this bovine alpha lactalbumin is very, very close to human alpha lactalbumin. And it has the least immunogenic potential within the body. That is why too much of research is also going on this bovine alpha lactalbumin functionalization, preparation of nanoparticles with respect to bovine alpha lactalbumin, and therapy associated with it. Apart from that, lysozyme is also present within the body. Lysozyme is also having antibacterial activities within the body. And this lysozyme can also be utilized for preparing these nanoparticles for targeted drug delivery. Since lysozyme is also present within our body, just you have to tweak with the immunogenic potential of that protein. And once it is done, you can utilize it for preparing nanoparticles. Now, these are the various sources of protein nanoparticles that we usually see. And we have got animal, plant protein, and human protein. Now, animal protein, I have classified them on the left-hand side. You can see gelatin, very commonly used animal protein for nanoparticle preparation. Collagen is also used for this nanoparticle preparation. We have this albumin for nanoparticle preparation. We have got casein. We have got silk protein. In plant protein, we have zein, we have gliadin, we have got soy protein. Soy protein is a very common type of protein available with us. And we have got human proteins. For example, human serum albumin, you have got lysozyme. These are the type of proteins we are having coming from the human source. Now, when we think, okay, we have finalized the proteins which we want to use for protein nanoparticles. Now, before going for synthesis and their characterization, we should be focusing on what type of properties we should focus on a protein. What type of properties can we improve? What are the properties we need to take care? We need to see those things. So before going into preparing this protein nanoparticle synthesis, we should consider what are the various properties of those proteins. So first we are going to look into the bulk properties. What are the bulk properties of those proteins? What is the solubility of the protein? Whether it is soluble in an organic solvent, whether it is soluble in a polar solvent, whether it aggregates in a particular solvent. What is the isoelectric point of the protein? How pure is your protein? Whether it is 99% pure, whether it is 95% pure, whether it is 100% pure. What is the molecular weight of that protein? What are the drug properties are there? What is the solubility of those drug molecules that you are going to attach to the nanoparticle of interest? What is the solubility of the drug molecule? Whether it is water soluble, whether it is organic solvent soluble, what type of organic solvent solubility is there? What is the log P value of the drug molecule? All these factors are going to play a very important role. Apart from that, the surface properties of the functional groups also are very, very important, particularly the charge on the functional groups. At what stage you have taken the protein for preparing the nanoparticle? What was the position when you have taken that protein? Whether it is folding, whether it is still unfolding, whether it is having hydrophobic reactions. All these things matter a lot before going into protein nanoparticle synthesis and doing their characterization. So we'll start with the first point with respect to the characteristics of the protein, and that is protein composition. Now, what is the composition of a protein? Usually, the protein nanoparticles come from various sources, and depending on the source from which the protein is derived, the protein will have different fractions. Now, the higher the number of fractions, it is going to have an influence on the nanoparticle characteristics. So, what are these fractions? Fractions in the sense, for example, we are using human serum albumin. Human serum albumin is having a free thiol groups that makes it capable to build dimers and higher aggregates. Now, initially, while preparing the nanoparticle, the different protocols are there. We'll see the protocols later. But I'll just tell you what are the steps are there so that you can understand why this different molecular weight fractions are created in the solution and why it hampers your nanoparticle preparation process. Now, when you are using a human serum albumin, you use it at a fixed concentration. Either you drop it into the organic solvent, or the organic solvent will come and mix with the human serum albumin. It can be done both ways, but the protocol says you have to use the human serum albumin and you have to drop into it your organic solvent. Now, whenever you are dropping these organic solvents are going to precipitate this protein. So as the solubility will decrease in the medium, the moment you are going to find out at that critical point the solubility of the protein is lost, immediately the protein will precipitate in that solution, and you will find white turbid color in that solution. That is a trigger point. You have to figure it out, and whenever that trigger point is coming, you have to immediately isolate whatever amount of protein nanoparticles are formed, because if you don't separate out those nanoparticles what are formed initially, later on whatever additional amount of organic solvent, for example, we use ethanol very frequently for preparing this protein nanoparticles. So these protein nanoparticles will be preparing a lot of molecular weight fractions. So initial whatever nanoparticles are formed, that is fine. You will be finding very less number of size of nanoparticles, and those nanoparticles will keep on building on each other, and they will keep on aggregating on each other, and you will be finding the size of those nanoparticles will keep on increasing. So human serum albumin is one of those proteins where you will be finding multiple fractions of this protein, and whenever you are going to break that protein or you want to precipitate that protein in the presence of an organic solvent, there is always a chance that you will be having this increased size of these protein nanoparticles. So this human serum albumin has a tendency to form dimers. So two monomers will combine together and they will be forming dimers. And the higher aggregates, as these dimers keep on building on them, or higher aggregates or higher particles of this protein come to each other, they will be forming higher aggregates. Now here, apart from these aggregates, sometimes some research protocols suggest the use of glutaraldehyde. Glutaraldehyde is a cross-linker which you will be utilizing for cross-linking those protein aggregates. So once the cross-link is formed, that cross-link becomes very, very stable, not easy to break, not easy to degrade. So at this point of time, you can add a drug molecule. The drug may be ethanol soluble, the drug may be water soluble. Depending on the choice, you want to use which type of drug molecule you want, water soluble or water insoluble, you can use it, not a problem. But problem comes when you have a specific amount of solubility in ethanol. Now we are using ethanol. All drugs are not 100% soluble in ethanol. So there will be certain point of concentration of drug that is soluble in ethanol. Only that portion can be loaded into that human serum albumin nanoparticles. Not only that, whenever you are using human serum albumin nanoparticles, particularly you are adding drug into it, there are two ways the drug can go into it. First one, whenever this aggregation process is going on and this glutaraldehyde is trying to trap all the amount of drug molecule. At that point of time, when you are adding the drug molecule, it is known as entrapment. You are entrapping the drug molecule within those protein fibers. So that is entrapment. Now, once your nanoparticle is formed and you are loading drug later on onto the surface based on adsorption principle, based on functionalization principle, that is called encapsulation. So you are encapsulating the surface of the nanoparticle with the drug molecule. So in protein nanoparticles, you can have both ways of drug delivery. One is entrapment drug delivery, another one is encapsulated drug delivery. Now, I'll give a very small tips at all the different stages when we are discussing about the characteristics of the proteins, so that you will have an idea that which way we should be heading in order to get a proper protein for our protein nanoparticles. Now, based on the composition of the protein, I'll give a small tip here. Always take the protein with the highest purity to get less polydispersity and uniform size nanoparticles. So whenever you are going to prepare a nanoparticle, my suggestion is go for the highest purity available with you. Highest purity you can get for that protein, go for it. Because you go for the impure protein. Suppose, for example, the protein is 95% pure, still 5% some impurity is there. That 5% impurities is going to ruin your total experiments just because those cross-linking won't be perfect. Higher aggregates will be forming. You will not get the desired size you are looking for. Suppose you are looking for a size range in 100 to 200 nanometer. Now your protein is not that pure, and you are following a protocol which utilizes a protein which is 99% pure, and you are using an impure protein. Definitely you're not going to get 100 to 200 nanometer particle size. Definitely the particle size is going to shoot up. 500 nanometers, 1 micrometer, go up to 2 micrometer, it can go up to 10 micrometers also. So thumb rule with respect to the protein composition is always take the protein with highest purity to get less polydispersity. Polydispersity means you'll get particle size in various ranges, starting from small particles to starting from large particle sizes. Small particles to large particle sizes, and all those particles are going to affect the protein size. So you'll be having less particle size, you'll be having large particle size proteins, and all this is going to affect the uniform size of nanoparticles. So whenever you are going for preparation of protein nanoparticles, always go for a pure protein in order to avoid polydispersity. So here I've taken these graphs from a paper written by Langer et al. In the year 2008, when he has prepared these protein nanoparticles. Here he has used different batches of this protein, human serum albumin protein nanoparticles. Now, as you can see, I want to show you something. You see the pH. pH is 7.5, and the highest pH we have is your 8.5. So 7.5, 8.0, and 8.5. You can see the graphs here. Whenever you are having a highly alkaline pH, somewhere around 8.5, you see the particle diameter. Now it keeps on around 150 to 200 nanoparticle. Now, if you see, if you are having a pH of 7.5, you are having a high particle size, and you see in pH 7.5, you are always seeing particles above 300 nanometers or above 250 nanometers. So alkalinity of the medium is also one of the important parameters while you are preparing the particles. Second most part, composition of the protein. Now the composition of the protein is going to decide the isoelectric point of the protein. Always remember, I'll come to that point also. Your window from the isoelectric point should be so huge that you can tweak in that window. Suppose your isoelectric point of the protein is 5.3 and we are working at a pH of 8.5. Then you have a better opportunity or better chances of getting a consistent nanoparticles at a pH 8.5 compared to 7.5, because pH 7.5 is close to the isoelectric pH of the protein, that is 5.3. So as you come close to the isoelectric value of the protein, you are going to get larger and larger sizes. Consistency in your protein nanoparticles won't be there. So as a thumb rule, always pick up the pH at which we are preparing the protein nanoparticles. There should be a huge window of difference from the isoelectric pH and at the pH where you are working. And at the same time, if you see the polydispersity profile of at different pH, if you see pH 7.5, this pH 7.5 is giving very high polydispersity, and the error bars, you see these error bars are very huge. If you see for 8.5, okay, here we have got a error bar somewhat close to this one, but if you see the polydispersity, it is comparatively low compared to the pH 7.5. And if you can see in this batch, here the polydispersity for 8.5 is somewhere between 0.025. And if you see the polydispersity here, it is somewhere around 1. So there is a huge difference coming in terms of particle size if you are preparing the particle size nanoparticles at a particular pH, like 8.5 or 7.5. Now, second point is your protein solubility. Now, how it is going to affect your nanoparticle preparation? Now, the solubility of the protein in the aqueous solvent or the organic solvent will also decide what will be the characteristic property of your protein. Now, a protein has a particular solubility in a particular solvent. Usually, most of the proteins you will be finding they are aqueous soluble. Okay, so whenever any protein is getting solubilized in a solvent, it has different stages of folding and unfolding. Now, what is this folding and unfolding? I'll give you one example. Suppose you are taking a raw egg. Okay, you drop that egg on the ground. Immediately you will see the egg white and the contents of the egg scattered throughout the floor. Do one thing, you take the egg at 37 degrees centigrade, you drop it. You find all the contents spilled on the floor. You increase the temperature of incubation from 37 to 45 degrees centigrade. Again, you drop it. Again, it will spill on the floor. You go to 50 or 55 degrees centigrade, incubate that egg and you drop it on the floor. Again, it is going to spill on the floor. Now you do one thing, take that egg, boil it in a pressure cooker, and once the egg is boiled, open the egg, drop it on the ground. Whether the protein is spilling on the floor? Now, why is this happening? And why it occurred up to 50 to 55 degrees centigrade? And when you boil the egg, it didn't happen. Now, proteins are always in a dynamic motion. Depending on the environment, the protein is going to change its structure. And whenever the protein is changing its structure, there are different components, secondary protein, structural components of the protein. The percentages are going to vary. Now you've got the alpha helix of a protein, you've got the beta sheets, beta strands of a protein, beta turns of a protein. Now this beta content, what is there, that keeps on increasing when you put too much of stress on the protein. So if you have a instrument where you can measure the alpha helix as well as beta sheet content, when you're keeping the egg at 55 degrees centigrade, you'll definitely find the beta content of that protein will increase compared to the temperature at 37 degrees centigrade. The reason is the protein tries to stabilize itself at extreme conditions. Now you're increasing the temperature gradually. Now under those increasing temperature conditions, your protein is trying to stabilize itself. Now whenever a protein will try to stabilize itself or try to protect itself, the beta contents of the protein are going to increase. Whenever these beta contents are going to increase, the rigidity of the protein is going to increase. Now, this is related to folding and unfolding. Now, you increase the temperature to 50 degrees centigrade for that egg. You allow it to cool down at 37 degrees centigrade. Again, that protein will refold back to the structure what is available at 37 degrees centigrade. So, immediately after 50 degrees centigrade, if you measure the beta sheet and alpha helix content, you will always find the beta sheet or the beta contents are increasing, but alpha helix content are reducing. Now, alpha helix content are always responsible for the flexibility of a protein. It helps the protein to change its structure. But whenever these beta contents are increasing, the protein loses its, what you can say, property of change of structure, depending on the environment where it is. So, and the final egg, boiled egg that I was talking about, it has totally lost its refolding capacity. The protein is folding and unfolding itself. Now, when you put it in a pressure cooker at a high temperature and pressure, the protein lost all its property to refold again to its previous structure. And what we see, a boiled egg is a irreversible form of the protein. Now that protein cannot come back to its natural state because already that threshold is lost. Beyond that point of time, we cannot go back to the initial state of the protein. So proteins are very, very flexible in nature. You cannot say this is the exact structure of a protein. That structure of a protein is specific for that temperature and that particular environmental condition. It may be a salt concentration, it may be a pH concentration, it may be a temperature. So protein structure is dynamic and it constantly changes over a period of time depending on the environmental conditions. Now, when we modify these environmental conditions of a protein, the protein will try to acclimatize itself with that kind of environment. So in that environment, if we make the environment conducive to increase the hydrophobicity of the protein nanoparticle surface, we can do it. We can increase the hydrophilicity of the protein nanoparticles, we can do it. Now, such type of modifications we can do only when the protein can unfold and refold again and again. Now, Duclair et al. In the year 2014, they have prepared this gliadin nanoparticles coming from a plant source, and he tweaked the solubility parameters of the protein, modified it slightly, and he demonstrated the size of gliadin nanoparticles could be controlled by its solubility parameters in both solvents used. So you're using a water as one of the solvent, you are using one organic solvent. If you control the solubility parameters in such a way, how it is precipitating in one solution, how it is precipitating in another solution, definitely you will be having the control on the nanoparticle size. Now, the smallest size of this gliadin nanoparticles could be achieved by solubilizing in the solvent that has the same solubility at that protein. So if you choose a solvent where the protein can go completely into that solution, always go for that kind of solvent for precipitating that protein, because you don't have any leftover protein that is still insoluble in that solvent. If you find that solvent, for example, water is a very good solvent, it can solubilize most of the water soluble proteins, and you have very less amount of precipitated proteins left in that solution. In those cases, precipitating such solutions will give you nanoparticles of very small size. Now, this is a very nice example I have shown you in this diagram. If you can see, this represents the pH value in which that protein is going to give you the protein nanoparticle. And if you can see, at pH value of 9, at the extreme corner, if you see the pH value of 9, you are getting the lowest size of nanoparticles. And at the extreme left, you are seeing the value at pH 7, we are getting the high size of protein nanoparticles. Now, this is due to the effect of pH. So, for example, human serum albumin has a pH of 5.05. So, at this particular point, the protein is not going to have any charge on the particle surface. So, the further you go away from the value of pi, and we are doing it the experiment at pH 9, you will get the least size of nanoparticles. Okay, and protein exhibits pH dependent water solubility. So at a pH away from the pi, the hydrophobic interactions will be reduced. So if you are using this pH 9, your hydrophobic interactions with the human serum albumin protein will be reduced, and consequently it will result in less aggregation. So because of that, you will be getting nanoparticles of very less size compared to pH 7, where you will be having a lot of hydrophobic interactions and your particle size will be very high, and your human serum albumin different fractions will be also there. So because of the contribution of all these things, your particle size at pH 7 will be very, very high compared to particle size at pH 9. Now the surface properties. What are these surface properties? How are they important to us? Now the surface chemistry of nanoparticles can be modified with the help of different functional groups addition to that protein. Now how we can functionalize the surface of the protein? You can get a drug molecule to functionalize the protein. You can have chemical functional groups that you can attach to the surface of the protein. You can add antibodies, you can add proteins, you can add peptides, you can add different linkers on the surface of the protein. And because by doing that, you modify the surface of the protein. Now, the surface of functional groups of the protein, for example, you have got amine group in a protein, you will be having carboxyl groups, you will be having thiol groups also on the surface of the protein. Now, these groups are exposed on the external surface. And whenever you use any chemical substance that is going to take advantage of these amine, carboxyl or thiol groups present on the surface of the protein, definitely you can do the functionalization on the surface of the protein in a productive way. Because most of the proteins will be exposing this amine, carboxyl or thiol groups on the surface of the proteins. Now, these surface amino groups are very, very important for a protein, particularly when you are preparing protein nanoparticles. And which is the cross-linker that is going to utilize this advantage? The cross-linker is glutaraldehyde. What this glutaraldehyde does is, it is going to interact with the surface amino acids present on the surface of the protein, and it is going to help in the cross-linking of that protein. Now, this glutaraldehyde is a bidirectional cross-linker. That means it can cross on one side with the protein. On the other side, it can cross-link with a drug molecule which is having an amino group also. So what it can do is, glutaraldehyde is a connection link between your protein nanoparticle, and in the middle you have got the glutaraldehyde. At the extreme end, you can have the drug having the amino group. So this glutaraldehyde as a cross-linker is very popularly used in most of the nanoparticle preparations. But in most of the nanoparticle preparations, the concentration of this glutaraldehyde is very, very high. Now, the surface of these amino groups can be further modified with the help of what? Hydrophilic polymers, for example, polyethylene glycol. Now, what advantage do you get by giving this polyethylene glycol to surface hydrophilic, increase the surface hydrophilicity? So what it will do is, it will increase the circulation half-life of the functionalized nanoparticle. Now, you want to avoid the phagocytic machinery present within the body, which is going to degrade your nanoparticle. You want to avoid it, functionalize it with a polyethylene glycol coating, or functionalization with a polyethylene glycol layer, and you will be avoiding this phagocytic uptake of degradation of this nanoparticle. Even more, the surface functional groups can be used for drug loading by electrostatic interaction. Increase the charge. Suppose you are doing this DLS particle size analysis, where you measure the zeta potential of the nanoparticle. If your zeta potential of a nanoparticle is suppose minus 29 or minus 30, at that level, no two nanoparticles will come close to each other. But the surface charge is so negative that if you give a drug molecule which is having a surface positively charged, immediately it will go and attract with the negatively charged surface, and immediately it will have an electrostatic interaction and it will be absorbed on the surface of the nanoparticle. So this surface charge is very, very important for electrostatic interaction between a protein nanoparticle and a drug molecule. So if you have opposite electrostatic charges, definitely there is going to be interaction. And this particular interaction is used particularly in the intestinal lumen when you are targeting the regions, particularly for irritable bowel syndrome, where you target the intestinal lumen in the colonic region. Now, here with respect to the surface, I'll give a small tip here. When the concentration of the cross-linker increases, you will get more and more denser particles. So your concentration, you increase the concentration of glutaraldehyde, the particle size is going to increase. Decrease the concentration of glutaraldehyde, your particle size will decrease, but up to a certain limit beyond which it will not form perfect cross-links. So you have to identify that point or that concentration of glutaraldehyde that is good enough to produce a proper cross-link in order to prepare a nanoparticle, but you are not overutilizing the concentration of glutaraldehyde for preparation of these protein nanoparticles. Apart from that, functionalize it with polyethylene glycol if required, if you want to avoid the phagocytic machinery of the body. Now, what kind of drug properties can affect nanoparticles? Now, there are certain nanoparticle protocols are there, where you add both the drug molecule, the protein, everything in one organic solvent or one aqueous solvent, and you straight away go with the protocol. There is a very less chance of really understanding what the drug molecule is doing at the protein surface. So physiochemical properties of the drug molecules are very, very important. How soluble is the drug? In which solvent, how much solubility is there? What is the log P value of the drug molecule? Whether that is good enough for binding with the protein nanoparticle? What is the molecular weight of the drug molecule? Whether it is more than the protein nanoparticle? How much protein nanoparticle you require for loading on the drug onto that nanoparticle surface? All these things matter a lot, and these things are going to affect the loading rate of the drug molecule on the top of the protein nanoparticles. You've got gelatin nanoparticles. You achieve higher encapsulation efficiency with respect to hydrophilic drugs compared to that of hydrophobic ones. So depending on the type of drug molecule, sometimes your protein nanoparticle is chosen, or there are certain nanoparticles where you can functionalize the surface depending on the type of drug you want. So usually gelatin nanoparticles are preferred for hydrophilic drugs. So water loving drugs will be attached on the surface of these gelatin nanoparticles, but it is not recommended for hydrophobic drugs. Also, the charge on the drug molecule is also very, very important, how it is going to interact with the protein nanoparticle surface. For example, you've got cationic doxorubicin, and if you want to load it on gelatin nanoparticles, it increases with increasing pH due to the achievement of the negative charge of gelatin at higher pH. So at higher gelatin pH, you will be having more amount of this negative charge, and it is going to attract this cationic doxorubicin on its surface, and there will be a perfect attachment of the drug molecule and the protein nanoparticle. Now, the tip here is you have to study the physicochemical property of the drug very carefully before you are going for the drug loading on the protein nanoparticle. You have to analyze the protein nanoparticle surface, what is the charge it is holding, what is the hydrophobicity it is having, whether your drug is hydrophobic, whether your drug is hydrophilic, what is the solubility of the drug molecule. All these physicochemical properties have to be dealt very carefully, particularly for drug loading on protein nanoparticles. Now, there are various methods of protein nanoparticle synthesis, and depending on the type of nanoparticles you want to produce, you can go for different types of protein nanoparticle synthesis methods. For example, you can form nanoparticles by increasing the protein unfolding. You keep on unfolding the protein, refolding the protein, and in between you add the solvent in such a way that you get the protein in a particular folding stage, and you will get a particular particle size of the protein. So there are different protocols are there where you can modify the protein unfolding and refolding, and you can decrease the hydrophobic intermolecular interaction in the chosen protein, and you can get the desired particle size at the desired condition of the protein. During the nanoparticle formation, lot of interactions keep on going on when the protein is changing its structure. Hydrophobic residues will come onto the surface, or if the hydrophobic residues are on the surface, they will keep on moving to the inner part of the surface of the nanoparticle. So amino acid exchange on the surface and at the center of the protein nanoparticle keeps on going on till it achieves a particular structure where it is going to precipitate, and you are using a cross-linker in order to cross-link those protein structures. Now, different types of methods are there. Desolvating techniques are there, concentration techniques are there, precipitation techniques are there, even high pressure techniques are there. All different techniques are there where you have to get this nanoparticle by solubilizing this nanoparticle initially in one of the two solvents. Either it is a water soluble solvent or it is the organic phase of that solvent. Apart from that, pH, salinity of the medium where you are performing this reaction, and temperature, all these things are important contributors how we are going to prepare the nanoparticle synthesis. So among all these methods, you have the first method that is known as the desolvation method. Very simple method, very easy to carry out. It is described by Weber et al. In the year 2000. And here the process is, you have a protein solution. Okay, if you can see at the beaker at the bottom, you have a protein solution here. It is under constant stirring. So you have to optimize the stirring RPM also, at what RPM you need to stir the protein. Otherwise, sometimes due to stirring itself also, sometimes it may precipitate the protein. So you have to optimize the RPM at which you need to stir the protein. Immediately, you are going to add the desolvating agent. Desolvating agent is your alcohol. Now, it is not that you have taken the test tube and simply you will pour the test tube contents into this solution, and immediately this nanoparticle is going to precipitate, and you will add the cross-linker and you will get the nanoparticles. It is not like that. You will take this desolvating agent and you will add drop by drop. Now, what are the different apparatus we have where we can give this alcohol drop by drop? Because this alcohol has a very low surface tension and it can immediately vanish from the tip itself. So always use syringes. 1 ml syringes are there, where whatever are used for this insulin injection, those kind of syringes can be used. You can use pipette tips that release 1 micro pipettes also you can use for dropping this alcohol. And while this protein is stirring on the bottom, you have to add this alcohol drop by drop, drop by drop. Now, depending on the pH, now you have to choose the pH also here. Suppose I am using human serum albumin, I'll prefer to take a pH of around 8.59 in order to immediately precipitate that protein. So pH will be also present in the solution, protein will also be present in the solution. Immediately when we'll be adding this alcohol drop by drop, immediately the protein will start to precipitate and it will form different particles, individual particles of those protein aggregates. Now you will be using a cross-linker. Now this cross-linker will be glutaraldehyde, and whenever you add this glutaraldehyde, all these nanoparticles are going to gel to each other and they will form a cross-link with each other. And that cross-link is immediately done at alkaline solution. Now, always remember, glutaraldehyde forms denser cross-links at an alkaline medium compared to acidic medium. So, if you are performing this reaction at pH close to 7, you will have a weaker cross-link. If you do it at pH 9, you have a stronger cross-link. So, that is the thing with glutaraldehyde. If you use it at a less pH, cross-links will be very fragile. If you do it in alkaline medium, your cross-links will be very nice. Now, once you do it, you can take these nanoparticles, go for characterization of those nanoparticles. We'll discuss about the characterization techniques once we discuss about this procedures, whatever procedures we are having. So, coming to the next process is the conservation method. In this conservation method, what you usually do is, you are going to look after the solvent polarity, pH, ionic strength, and the presence of electrolytes. So here in conservation technique, you have a liquid-liquid phase separation. Means two liquids will be there, but there will be a phase separation of one of the protein in one of the liquid medium where it is easily going into that solution. So two liquid phases will be there. One salt will be added to it, and that salt is going to provide an environment where the protein is going to precipitate out. Now, this method has been proposed by Elzobi et al. 2015, and if you can see the figure here, you have the solvent and you have the salt also added to it, and you are stirring it, the protein precipitates, and during the process of stirring, you add the glutaraldehyde cross-linker, immediately you get the cross-linked nanoparticles. Now, what is the additional thing you have here? Apart from anhydrous ethyl alcohol that you are adding, that is at a protein concentration will be somewhere around 150 ml BSA, where 5 mg per liter, you have tris hydrochloride salt and sodium azide. This is the chemical substance that is going to precipitate the protein in addition to alcohol, in addition to the aqueous solvent. So this salt is very, very critical. Tris hydrochloride along with sodium azide, very, very critical for precipitation of the proteins. So this is another method, conservation method. And in this conservation method, you are going to precipitate the proteins in the presence of a salt. Next method is the emulsification by solvent evaporation method. Now, what is this emulsification by solvent evaporation method? Now, in this method, we utilize high-speed homogenization process. That means you will be using an ultrasonicator at a much higher wattage, so that sonicator vibrations are going to create those fragments in that protein structure when you are adding this aqueous solution in a non-aqueous phase. So there are two steps in it. Sometimes people do up to one step cross-linking only. In certain conditions, people will go for 2 phases of cross-linking, but the process is same. Just you have to repeat the ultrasonication process 2 times. So first you will be preparing this water by oil emulsion. You have the oil phase is there, and you have the protein solution is there. You have to drop this protein solution. Along with that, sometimes people will be adding this drug molecule into aqueous solution, or drug will be in oil solution. So when these nanoparticles will go into solution, they are going to break into aggregates. Now these aggregates along with drug molecules will be there. You give a cross-linker, immediately the drug particle entrapment will be there, and that cross-linking process will be done with respect to the drug molecule. If you want to go for the second step, again you can go for this ultrasonication, where you will be using these aggregated drug particles along with the drug molecules. Simply go for the ultrasonication process. It is going to segregate if at all any protein aggregates are more. If it is there, that is going to break it off. It is going to come in contact with the drug molecules. Now those drug molecules will come to each other with the protein nanoparticles depending on the charge they are having on the surface. A protein nanoparticle having a different charge, drug molecule having a different charge, they will come to each other, and during the process you add glutathione. And so that is going to cross-link the thing, and you are going to get the nanoparticles. So that is by emulsification by solvent evaporation method. Next one is your nanoprecipitation method, and that is proposed by Fessy et al. In the year 1989. Now what it does is, it is based on solvent displacement method. Now it requires 2 immiscible solvents. One has to be your aqueous solvent where a protein will go. Another has to be a non-aqueous solvent, maybe an organic solvent, maybe a oil solvent. We'll go for the organic solvent, for example, ethanol. Now here, the drug-loaded nanoparticles from hydrophobic proteins can be prepared. For example, zine and gliadin are used for this method. Now here, what you do is, you have a organic solvent. This organic solvent is ethanol. You have to add drop by drop. Here you have got the protein solution where the protein is already there. You are giving this ethanol over here so that it is causing aggregates in that protein. And in the process, what is the external solvent will be there? That external solvent is going to get evaporated. So what is this blue solution you are looking here? That blue solution will be evaporated, and finally whatever you will be left over will be with nanoparticles only. Now one of the demerits of this method is, when you are evaporating the solvents, you are treating the samples with temperature. Now this temperature can have a effect on this protein nanoparticles, even on the drug. So this is the one which is the least preferred nowadays. We don't use this method for preparing these nanoparticles. Next method, and one of the most useful methods that is used when you are going to prepare on a larger scale of nanoparticles. Now you want to prepare industrial scale nanoparticles. So at such a large scale, you have to do it in a nanospray drying. This is nothing but you are going to have a concentration of this nanoparticle solvent where it has precipitated. Along with that, you will be having an organic solvent phase and the aqueous solvent phase. Both will be mixed. And they will be inserted into the chamber with the help of a nozzle at a very high speed. So whenever the particles are coming out of this nozzle with a very high speed, and they go into this drying chamber, the particle size will be less, and immediately it dries out. So immediately when the particle dries up, it will be having certain charge on its surface. Now, depending on the charge on its surface, the particle is going to move to the positive side, or it is going to move to the negative side. And here collectors will be there. Those collectors are going to collect the charged particles, whether positively charged particles are there or negatively charged particles are there. Now, the good thing about this one is you can control the particle size based on the spray mesh size. You have a very small spray mesh size at the nozzle, You just reduce the size of the mesh, you will get less particle size. You increase the size of the mesh, you get large particle size. And surfactant concentration. You will also have a surfactant that lubricates the movement of the particles within this chamber. So surfactant concentration is also very, very important in preparation of industrial scale preparation of nanoparticles. Next, we'll come to the characterization. Now, in characterization, you basically characterize the size. In characterization, the major problems come with the particle size analysis. Now, here I am showing you the size of a scanning electron microscope image. Now, the scanning electron microscope image has certain demerits. What are those demerits? One is your resolution, one is your sample damage, third one is your sputtering issues. Now, what is this resolution? Now, in scanning electron microscope, as you increase the resolution, there is a high chance that you are going to damage the sample, because the energy that is being thrown at this nanoparticle, these nanoparticles are not going to handle that much amount of energy. Before you can take any image, even your sample can get destroyed. So resolution is very demerit of this scanning electron microscope. You get your sample damaged. And sputtering, what is the sputtering issue? So you have to coat these nanoparticles with a certain conducive particles, so that whenever the charge is thrown on it, the charge dissipates very nicely through this conducive particle and you get a very nice image. So if you sputter the surface of the nanoparticles with a element, suppose for 40 second, 50 second, the coating will be so used that you cannot find the edges properly. Okay, so sputtering time is also very, very important when you are analyzing this scanning electron microscope images for size. And here, if you have too much of sputtering time, you will lose the edges of these nanoparticles, and these extra sputtering will be depositing in between the grooves of this nanoparticle, and you won't be getting a perfect size of a scanning electron microscope. Next we have is a field emission scanning microscope. Now I'm not going to discuss on the protocol. This is a general protocol. You can find it in any of the articles. I'm just going to look into the problems you are going to face in this field emission scanning electron microscope. Now this one is also very nice images of protein nanoparticles you can get. But again, here one of the problem is at the background, what you see is a carbon tape. Now this carbon tape is used to attach to the silicon wafer, and that carbon tape should be attached to the aluminum stub at the bottom. Now these are small things. If you miss out, if this carbon tape is not connecting to the aluminum stub at the bottom, the charge is not going to get transferred from the surface onto that aluminum stub. And in that case, you won't be getting a perfect image on the surface. So that is a minor thing that you should be knowing, that this carbon tape should be attached to the aluminum stub at the bottom. Unless and until you do it, the charge dissipation is not proper, and whenever you are taking the image will be very blurred. Sometimes you can damage your samples when you are taking the images also. Moreover, it requires very less voltage. As you can see, 5 kilovolt energy is only required, and that is why the chances of damage of the sample is very, very less. Whereas in case of SEM, if you see, you require 10 kilovolt is required. Somehow these researchers, they have got a very nice pictures at 10 kilovolt. But as far as I have seen, at 10 kilovolt, when you are doing a scanning electron microscope and you go for a higher focusing image, you lose your sample. Next is your atomic force microscopy. Now this atomic force microscopy comes into picture when you want to see the three-dimensional view of your nanoparticle. Now I have shown you here only the two-dimensional image of our picture due to limited space availability. So I have taken this image from this article published here in Biointerfaces. So here you can clearly see this drug molecule coating on the nanoparticle surface. Now the image for the 3D also, you can get it from the sidewise images where it will be clearly elevated from the surfaces. So whenever you go for atomic force microscopy, you are looking for 3D images of the particle, where you need to see what is my drug coating on the nanoparticle surface. Then you can easily see these are the nanoparticle drug molecules on the surface of the nanoparticle. Now, some places this drug coating has been removed, some places it is consistent. So just for a representation purpose, I have shown you. So atomic force microscopy will give you three-dimensional image of the particle. Now we come to the DLS particle size analyzer. Now, if you see the two images, what you see here, one is giving you the zeta potential, another one is your particle size. Now I got a particle size somewhere around 200 to 300 nanometers. You can see the peak over here. And I've got a zeta potential of somewhere around 30 millivolt, So when you are having a charge of minus 30 millivolt, the chances of aggregation of particles will be very, very less. So always try to achieve maximum zeta potential that can be obtained from your nanoparticle. The more it is, the better it is. So the chances of aggregation of your nanoparticles will be less. So the peak, if it is close to zero, you will be having more amount of aggregation in such kind of nanoparticles. Moreover, whenever you are doing this nanoparticle analysis, particularly with respect of DLS or the dynamic light scattering analysis, if the surface of the nanoparticle is hydrophilic, be careful. And the solvent you are taking for DLS analysis, be careful. If you have a hydrophilic surface, the hydrophilic surface is going to add water molecules and the nanoparticle is going to swell. So in those cases, you will have a more bigger size of nanoparticle you get in the water medium. So in those cases, you have to replicate what is the physiological concentrations of all the proteins in your plasma, prepare a similar looking solution, and then you can go for a DLS particle size analysis. Next, you should be doing your FTIR analysis. Now, with respect to FTIR, with respect to a protein, you are looking for these two amide peaks, 1600 to 1700, 1510 to 1580. And these are the regions where you will be frequently finding this peaks in a native protein. So NMLYZ is nothing but native lysozyme, and SN is nanostructured lysozyme. So you see in native lysozyme, you will be finding this prominent peaks of these two amide peaks in the native lysozyme. But whereas in case of nanoparticles, due to cross-linking of these amide bonds in the protein, you won't be finding these two bands, and rather you will be finding one small band, which is around 1605 centimeter inverse for lysozyme. So this is for a representative purpose only. You can also characterize with the help of FTIR analysis, and you can observe this amide 1 and amide 2 bands. Next, you have to go for CD spectroscopy. What is this CD spectroscopy does is, I have told you previously about alpha helix content of a protein and beta sheet content of a protein, right? So when you put a protein in a stressful environment, your beta content will increase. Now, nanoparticles will always have a increased beta content. The reason being, you are precipitating the protein, and always the protein tries to be in a stable structure. But under the influence of the solvent, the protein precipitates and you get these protein nanoparticles. So whenever you have these protein nanoparticles, you will always see both the alpha helix content and beta sheet content are going to increase in such nanoparticles, whereas the native lysozyme will always have less. So what it does is, alpha helix content will be at the center, beta sheet contents will be at the periphery, which are very rigid in nature, that provides increased amount of stability. So this CD spectroscopy analysis will tell you about the secondary changes in the structural components, particularly alpha helix change, beta sheet change, and that will give you an idea about the rigidity of the protein structure. Next, you can do fluorescence spectroscopy analysis, where you can do a normal fluorescence spectroscopy analysis, where here I have done with lysozyme. In lysozyme, you will be finding the peak around 350, but in case of nanostructured lysozyme, due to the loss of these tryptophan residues, the tryptophan residues come to the core of the nanoparticle structures, and because of that, you will be finding there is a decrease in intensity of the nanostructured lysozyme. Whereas you see ANS fluorescence intensity, that is another type of fluorescence spectroscopy, which you do to measure the surface hydrophobicity. So nanoparticle surface will be having more amount of surface hydrophobicity. For my case, it was very high hydrophobicity, and native lysozyme is having very less hydrophobicity. So advantage is, in lysozyme nanoparticles, I can load more amount of hydrophobic drugs. Now, coming to the conclusion, these nanoparticles are proving to be a breakthrough innovation in the field of pharmaceuticals. And I see in the next 10 to 15 years, most of the formulations will be having some form or the other form of the protein component for targeted drug delivery. Moreover, they are biodegradable, and unnecessary undesirable metabolism is also not there. So that is an advantage for these protein nanoparticles. Now, these protein nanoparticles have unique properties compared to other nanoparticles. Some I have discussed previously: easily available in nature, easy to manipulate, easily biodegradable, non-toxic, less immunogenic. So these are the advantages of these protein nanoparticles, and you can functionalize them with n number of substances, and you can target your target site. So with my point with respect to the synthesis and characterization of protein nanoparticles is, the choice of the preparation method is very, very critical. Which preparation method you are choosing, which protein you are choosing, what are the characteristics of the protein, what is the characteristic of the drug molecule, what is the encapsulation efficiency, what is the delivery target, what is the therapeutic approach. All these things play a very important role how you are going to prepare the nanoparticles. Okay, so this is about my presentation.
Thank you, sir. Thank you for your wonderful presentation, sir. Now I request you to address some of the queries raised by our delegates.
Okay.
Sir, here is the first question, sir.
Yes.
In solvent evaporation, conservation, nanoprecipitation, and in other methods, we generally get spherical shaped particles.
Yes.
How does this particular shape is formed? And what would be the required parameters to control the shape?
You are using a stirring process, right? In this, all these methods, if you have seen, you have kept a stirrer at the bottom. The process of that stirring stirrer is nothing but to polish the surface of the nanoparticles. So if you don't use a stirrer and you prepare nanoparticles, you will observe your nanoparticles are of irregular shape. You won't be finding a particular shape. The spherical nature of the particles come from the stirrer. By adjusting or monitoring the stirring time or the stirring speed, you can regulate the size of the surface of the nanoparticle also. Less stirring time, more particle size will be, size of the particle will be more. Higher stirring time, particle size will be less. But there is, you have to optimize it. If you put it at very high stirring time, sometimes the particles will be damaged and you will have an irregular surface. So you have to optimize the stirring time based on the nanoparticle you are taking into consideration.
Sir, here one more question, sir. The temperature can affect the nanospray drying method.
Yes, definitely. Temperature is one of the factor. That is why you have to control the ambient temperature at which you are releasing this nanospray thing. And you should be very, very careful with respect to drugs which are prone to degradation in high temperatures. Not only that, you should be choosing a protein which is stable at that high temperatures also. So temperature will also denature the protein, and the cross-links between the protein and the drug molecule is also getting compromised at very high temperatures. So you have to reduce the temperature by reducing the ambient temperature, or else you have to look for a protein which is stable at higher temperature. Look for a drug molecule. You cannot just change the drug molecule. Better look for a protein which is stable at a higher temperature.
Sir, here is the third question, sir. We could able synthesize different shapes of okay, sir, already that was over. No, sir. If we take new protein for nanoparticle preparation, what are the key areas we should focus?
Okay, now this is a very good question. When you are choosing, you don't know anything about that protein, what kind of information you should collect about that protein? First is, know the isoelectric point of that protein. Identify it. Why I'm saying it is important is, if you know the isoelectric point of the protein, just you increase the window where you are going to prepare the protein nanoparticles. Suppose, for example, human serum albumin. It has an isoelectric point at 5.03. If you are preparing a nanoparticle at pH 7, as I showed you in one of the slides, you are not going to get smaller size nanoparticles. Rather, you will be getting microparticles. So move away from the isoelectric point of that protein to an alkaline region where you will be getting narrow size of the proteins, and because of the less amount of hydrophobic reaction. Second important point is, choose a protein which is available in human system. Why I am saying this is because most of the proteins coming from plant origin or animal origin, there is an immunogenic component involved with it. You cannot use those proteins directly into human beings. So my choice is, pick up a human protein that is available in the body, get it recombinantly produced in some organism, maybe E. coli. You can go for a plant-based production also. And using that recombinant protein, you can go ahead with the nanoparticle preparation. Third one is the surface hydrophobicity of the protein. That is very, very important. Now you are loading a hydrophobic drug, you want high surface hydrophobicity. Check your nanoparticle through ANS fluorescence studies, how much hydrophobicity of the surface you are having. If you are having good surface hydrophobicity, you can load a hydrophobic molecule. But without checking the surface hydrophobicity, simply based on electrostatic interaction, you want the drug molecule on the protein surface, it is not going to happen. So once you prepare the protein nanoparticles, you have to measure the surface hydrophobicity. If it is high, go for a hydrophobic molecule for putting it on the drug surface. The next point is, what type of drug you are using for that protein nanoparticle loading. Whether the drug molecule is having proper interaction with the protein, whether there is sufficient functionalization available on the surface of the protein, that is also critical for the preparation of this protein nanoparticle. And initially, the usage of glutaraldehyde, that is also very, very important. Most of the protocols show they use 25% glutaraldehyde, 10% glutaraldehyde. Some papers have also mentioned 40% glutaraldehyde. But remember it, higher concentrations of glutaraldehyde itself is toxic to cells within our body. Now, what I have found is percentage of glutaraldehyde less than 1% is very, very suitable for preparing good enough cross links for nanoparticles without producing toxicity in normal cells of our body. As you increase the glutaraldehyde concentration, you may get a very good nanoparticle, but the cross links will be there that contain glutaraldehyde, and that high concentration of glutaraldehyde is toxic for our body. So hence avoid a high concentration of glutaraldehyde. So these are some of the things you should focus before choosing a protein nanoparticle.
Sir, one more question, sir.
Yes.
How much percentage of purity of the protein can maintain during design of protein nanoparticle?
My suggestion is always go for 99.99% protein. But if it is not available, you don't have a choice, then you cannot do anything. My suggestion is always go for the purest form of the protein. Unless and until you have the purest form of the protein, you will definitely going to get more or high particle sizes. You cannot get consistently low particle sizes. See, it is the impurity that affects the particle size integration into that nanoparticle. More amount of impurity, more amount of increase in your particle size. Less amount of impurity, you can tweak with the protocol, increase to the alkaline pH, and you can prepare the nanoparticles of a lesser particle size.
Sir, here is the another question, sir. How does the fluorescence microscope can determine the hydrophobicity?
It is not the fluorescent microscope, it is fluorescent spectroscopy. So in fluorescence spectroscopy, there are two types of fluorescence you can study. One is tryptophan fluorescence, that will give you the overall tryptophan residues present on the surface. So whatever fluorescence intensity you are getting, it is due to tryptophan that is present in the surface. Now, if tryptophan is going into the core of the nanoparticle and you are measuring the tryptophan fluorescence on the fluorescence spectroscopy, you won't be getting any tryptophan fluorescence. That is clearly an indicator that your tryptophan residues, whatever are present on the surface of the protein nanoparticle, are going inside the nanoparticle core. Surface hydrophobicity can be measured with the help of that chemical compound, that is ANS. Okay, that is ANS is anyl sulfonic acid. That component is used for measuring the protein surface hydrophobicity, and this particular chemical substance will bind with the surface hydrophobic sites on the surface of the nanoparticle. So a particle, nanoparticle, which is having high surface hydrophobicity, you will always get a high ANS fluorescence. So if your ANS fluorescence is low, that is an indicator that your surface hydrophobicity is low. So never go for hydrophobic drug loading on that type of nanoparticle. Any nanoparticle having high ANS surface fluorescence, definitely it is having high surface hydrophobicity. So you can take any hydrophobic molecule and you can go ahead with the loading of that nanoparticle.
Sir, another question, sir. Most of the proteins can degrade at low temperature. So how to regulate the degradation rate of proteins while preparing?
Now, this is a very good question. Now, I have seen a lot of students face this problem. Suppose, for example, human serum albumin that comes in a pack size of 100 mg. So what we observed during our initial days of nanoparticle preparation is we require only 1 mg of that protein. So what we do, we take the protein box, we take out 1 mg of the protein, prepare a sample, go ahead with the nanoparticle preparation process. We get it fine. The particle sizes are fine. But during the process, what happens is atmospheric air has entered into that box and it is already reacting with the protein that is already available in your box, which you are thinking of using in your future experiments. So by that time, when you are using the second lot of protein, already there is some degradation happened in the protein. Even if you keep the protein at 4 degrees centigrade also, there is a constant rate of degradation in the protein. So you cannot stop the degradation of the process. But my suggestion is, when you get the protein vial of your interest, immediately solubilize it into your solvent and go ahead and prepare take the entire stock of that protein, prepare the nanoparticles. Don't wait for another batch to continue, subsequent batches to continue. By that time, your protein is going to degrade and there will be inconsistency in different batches of your nanoparticle production. So whenever you are preparing protein particularly, pick up protein how much you can handle in your day-to-day experiments. For example, you are going to prepare 100 mg starting material as a starting material for protein nanoparticles. Just buy the 100 mg pack size only. Don't buy a 500 mg pack size and you are going to use only 100 mg. So whenever you are going to open the vial of that protein, use it completely, prepare your nanoparticles, then you store it. But if you buy the protein, take small amount of it and do the protein nanoparticle preparation, definitely on storage, your protein consistency will be lost. Do we have any more questions? Do we have any more questions?
Yes, sir. No, sir. Sailendra, sir. Thank you very much, sir.
Okay.
So, thank you very much, sir. It's a very valuable. So thank you very much, sir. It's a very highly valuable and informative session. So I request all the participants to implement these valuable things and informative things for your future growth. On behalf of AICTE ATAL FTP and Nirmala College of Pharmacy, I request the Dr. Sailendra Kumar Mahanta to accept a token of gratitude to receive the e-certificate.
Thank you, sir. Thank you for the opportunity on this ATAL FDP platform.
Thank you, sir.
Thank you,
Thank
sir.
you, sir. Thank you all participants. So
Thank you all the participants. Thank you all.
Dear participants, you can join the session 2 at 12:05. We'll share the YouTube and Zoom link in our WhatsApp and email. Thank you. Thank you, one and all.